Okay. So on Wednesday, we started talking about synapses, and that's what we're going to continue with today. I want to do a quick review of what we went over on Wednesday, because I know a lot of people kept getting kicked out of the meeting, and I took too many cold meds, so I don't feel like what I said was necessarily the clearest. So I'm going to give you a rapid fire summary here. Okay. So really what we were focusing on, the new material, was about propagation of the action potential, and then about what's happening with neurotransmitters at the synapses. So this is a big diagram of an unmyelinated action potential being propagated down the axon. So when we're talking about the action potential, we're talking about that depolarization to positive 30, which kind of fires off all or none. And we can see that what actually happens to get it down the axon is that basically it's progressively triggering further and further sections of the axon. So like as one region depolarizes, that charge slightly diffuses to the region next to it, which then triggers those voltage gated channels. So they do that whole same spike in progression down the axon. So that's all this diagram is showing you. Is if you want to go through in detail, you can see when the positive charges flow in, then we have arrows showing that they're spreading out, and then we can see that the next little bit of the axon then fires off the action potential. So we think of this all as one action potential that's moving down the axon. So we wouldn't call this like 3 action potentials when A, B, and C fire off. It's just one action potential, but it's going progressively down the membrane. Okay. And we compared this to conduction in a myelinated neuron, which is a little different. And specifically, myelin makes neurons conduct these action potentials faster because they don't have to trigger every bit of the membrane. They just kind of jump between gaps in the myelin. So myelin wraps around our neuron, covers over the membrane here, and we have these nodes called nodes of Ranvier. So those nodes of Ranvier are where you'd see concentration of your voltage-gated channels, and that is where you would see the action potential. So it would go directly from one gap in the myelin to the next gap in the myelin, not having to trigger this stuff in the middle. So this would conduct an action potential faster. We call it saltatory conduction. There is one other way we can make action potentials move faster, which is to make a neuron wider, basically. Just speeds the flow up of these charges if we have a wider tube, basically. So an increased diameter if we're talking about it formally. Okay. And you have a similar image that you can look through for a myelinated axon versus an unmyelinated one. So here we can see that in our myelinated axon, whenever we see the yellow, that's telling you that's where the action potential is in these diagrams. So this yellow, you can see the positives inside, indicating that portion of the neuron is depolarized at that moment in time. And then this is our next moment in time, and we can see that that depolarization jumped. So it jumped and triggered the depolarization of this second portion of exposed membrane in the node of Ranvier between the myelin. Okay. So any lingering questions about propagation here down the axon? Okay. So then our second topic on Wednesday was about our presynaptic and postsynaptic neurons and the synapse more generally, which is the space between these neurons, basically. So synapse is when we have one neuron signaling to something else, and really what we're talking about here is neurons signaling to neurons. We'll talk about neurons signaling to muscles, for example, in our next unit. Okay, so we can name what part of the postsynaptic neuron is receiving the signal. So we can say that the synapse is occurring between the axon of our presynaptic neuron, and the dendrite of our postsynaptic neuron, the axon of our presynaptic neuron, and the soma, the cell body of our postsynaptic neuron, or the axon of our presynaptic neuron and the axon of our postsynaptic neuron. And that one is especially important. We're going to get to some more details on those axonic synapses today, because they work a little differently, and these are really where we're going to see modulation of the release of neurotransmitters, whereas for the axodendritic and axosomatic synapses, those are basically going to be producing graded potentials, which we talked about last week. Okay. So we had our two types of synapses, our electrical and chemical. Here we can see presynaptic neurons, so like neuron number 1 connecting to a postsynaptic neuron, neuron number 2. So we have two ways that we can signal here in these synapses. The first one, which we talk about less, is an electrical synapse. So that's when the two membranes of the presynaptic and the postsynaptic neuron press directly against each other, and we form a gap junction between them. So that's a direct path connecting the cytoplasm. So any ions or second messengers or whatever that's in the cytoplasm of one neuron will get into the cytoplasm of the next neuron, and vice versa. A gap junction, in case you didn't talk about them in your prerequisite courses, you can kind of think of them like ion channels. They're not the same. They're connecting two cells. They're bigger, but they work kind of similarly. When they're open, they're just open and stuff flows through. Our chemical synapses are trickier. They're slower because we have to trigger the release of neurotransmitters. Then the neurotransmitters have to exit the cell through exocytosis. Then they have to bind to a receptor on our postsynaptic neuron, and then they'll get cleared away eventually. So this is what we're going to spend more time talking about. The chemical synapses, which are slower, and they just go in one direction because we have one neuron, the presynaptic neuron, signaling to our second neuron, the postsynaptic neuron. Okay. So we saw the steps. I'm going to say these super fast. Our action potential, our depolarization, comes down to the axon terminal. Once this region, this axon terminal, depolarizes, so it spikes to positive 30, this opens voltage-gated calcium channels. Calcium flows into the cell. When there's calcium in the axon terminal, that tells this neuron to release neurotransmitter. This happens when these synaptic vesicles, which are full of neurotransmitter, bind onto the membrane, break open. This is a description of exocytosis. They release the neurotransmitter into this area we call the synaptic cleft. Once we have neurotransmitters in the synaptic cleft, kind of two things are going to happen to those neurotransmitters. We're going to track what's happening to the signal part first. So once we have these neurotransmitters here, number 4 in the synaptic cleft, they bind to receptors on our postsynaptic cell. So they don't go inside our postsynaptic cell, they're just binding on to proteins in the membrane. But then that receptor, which is a protein, has an internal part as well, which creates some kind of response in the cell. And we'll look at two types of responses today. After a while these neurotransmitters are floating around in the synaptic cleft, they're going to come into contact with some other proteins. One of those is enzymes on the membrane of the postsynaptic cell, which degrade the neurotransmitter, so they break it down. So then those broken down parts diffuse away into basically the soup that our neurons are in. We saw like CSF in the brain when we were in anatomy lab. So they'd move into that fluid basically. The other thing that's going to happen with those neurotransmitters is some that don't get broken down are going to connect here to our reuptake molecules. So that's number 7 here, which means that our presynaptic cell is going to basically reabsorb those neurotransmitters so they can use them again, basically. And the way some drugs work, like our SSRIs, is basically that they kill this number 7. They're reuptake inhibitors, which means that you have neurotransmitter floating around in the synapse more. So it basically acts like you have more neurotransmitter total because those neurotransmitters spend more time in the synapse. So like they can respond to more receptors because they're there a little longer. Okay. Now we're going to move forward. So questions about this flow of steps from Wednesday. Okay. So a thing to know about these chemical synapses. So these are the steps specifically for signaling at a chemical synapse. So you should associate chemical synapses in your mind with neurotransmitters. We don't see those in our electrical synapses. The other thing we know about chemical synapses is that they're a little slower than the electrical synapses. So in the electrical synapses, we had ions basically flowing back and forth across the cytoplasm, just like that. We don't have to do anything else, just open that gap junction. In our chemical synapse, there's a little bit of a delay. These are slower because once the action potential arrives to the axon terminal, it's not going directly into the postsynaptic cell, right? Like we don't have this propagation wave, just like action potential here, more ions, action potential here. We have to do all these steps in order to get neurotransmitter into the synaptic cleft. So basically what we're saying is this is slower because it takes a little while for the calcium to get into the axon terminal, cause this vesicle docking, which is how we're dumping the neurotransmitter. And then that neurotransmitter has to actually be released. It doesn't take any really significant amount of time for neurotransmitter to diffuse here. It's really this delay is about this process we have to go through to release the neurotransmitter in the first place. Okay. When we're talking about fast and slow, we said that the electrical synapse is fast, chemical synapse is slower, but we can then also break down chemical synapses into a faster type and an even slower type. Okay, so that's what we're going to do right now. So right now, basically the difference we're looking at is, I'm going to scroll back for a second. We're going to look at different types of receptors here. So this purple box. So what type of receptor we're attaching to makes a difference for how fast the signal gets through. So one type of receptor, and this is going to be on our postsynaptic cell, right? This is the cell that's receiving the signal. So on our postsynaptic cell, one of the types of receptors we'll see is something called a channel-linked receptor. And we've actually kind of seen this before, like when we were talking about graded potentials. I think that was last week, might have been a couple weeks ago. But when we were talking about our graded potentials, we talked about ligand-gated channels. And this is basically what we're talking about. So in this case, our neurotransmitter would basically be our ligand. It binds on to a receptor that is itself an ion channel. So it is a receptor, but it's also an ion channel. So what happens when this ligand, this neurotransmitter, binds onto this channel is that it opens, and then we just have the direct movement of ions through that channel. Okay, so this quickly changes the membrane potential in our postsynaptic cell because ions moving, diffusing from extracellular fluid in the synaptic cleft to the intracellular fluid in the cytoplasm is fast. And when you have a channel open, that's just kind of a fast process because our ions move down their electrochemical gradients. Happens quickly, just like with our action potential when we open voltage gated channels and those sodium ions flow in really quickly. The same kind of thing happens here with our ligand gated ion channels. The other thing about these channel linked receptors is that as soon as the neurotransmitter leaves, this channel closes up. So if the neurotransmitter's gone, the channel is closed and the signal stops. So it happens fast to get the signal through, but it also happens fast to shut off the signal. So it's fast kind of in both ways. We have a specific word we use for these channel linked receptors opening ligand gated ion channels. We call this ionotropic receptors. So a way to remember this is this ion part in the beginning I find helpful. For opening ion channels, we have an ionotropic receptor. I should say if we're directly opening ion channels, because we'll see that they're involved in our other types of receptor, but there's more steps. Questions about these ionotropic receptors? So we're going to compare these ionotropic receptors to some G protein coupled receptors that we will also refer to as metabotropic receptors. So our fast channels are the ionotropic receptors. These are metabotropic receptors. So those are like opposites of each other. In this unit, we have a lot of words that are opposites, which is why I'm highlighting that. Okay. So this is our slower function of neurotransmitters. So if you've looked at G protein coupled receptors before, hopefully you remember a little bit about how they work, but we're going to describe it in brief now. Basically, a G protein coupled receptor, just like our previous receptors, just like our ionotropic receptors, starts to work when a neurotransmitter binds to the receptor. But a G protein coupled receptor is not an ion channel itself, really. A metabotropic receptor, a G protein coupled receptor, has on the inside of the membrane, on the intracellular side, something called a G protein. So this is basically a peripheral membrane protein. Remember we talked about integral and peripheral membrane proteins. This would be an example of one of our peripheral membrane proteins. So this G protein that's attached to our G protein coupled receptor, when we have the neurotransmitter bind on to the receptor, this G protein breaks apart and it kind of travels down the membrane a little bit. When this G protein breaks apart and travels down the membrane, its various parts, specifically here this alpha part, then activates or inhibits something else. And I'm saying something else because we actually see two types here in our two diagrams. Okay. Both of these are metabotropic receptors, though. They're both metabotropic receptors. They're both G protein coupled receptors. We're just having a slightly different cellular response to this G protein. So in this first image, which is the harder one, the G protein is activating this enzyme. And this enzyme, its job is to produce a second messenger. So we can think about the neurotransmitter as the first messenger outside the cell. The second messenger inside the cell hangs out inside the cell. Cyclic AMP or CAMP is a common one. Just in case you see that in the future, we do sometimes see that word. So that's a second messenger. So something this cell made. So this is now hanging out in the cytoplasm. And then the cell has a lot of sort of secondary responses to that molecule that we've made to that second messenger. One of those responses will be, if we're talking about these neurotransmitters, to open or close some kind of ion channel. So we, like I said, we do eventually get to an ion channel. It's just a lot more complicated than with the ionotropic receptors. So the ionotropic receptors, we just immediately open the ion channel because the receptor was an ion channel. here with our metabotropic receptors, we have to bind to the receptor, release this G protein, potentially make a second messenger, which then indirectly, right, we've opened this ion channel. The second messenger can have other effects in the cell as well, in addition to opening these ion channels. So we'll see actually that cyclic AMP does a lot of different things in cells. Okay. Our easier type of G protein coupled receptor here can more directly open the ion channel. Still not as direct as with our ionotropic receptors, but we do have some G protein coupled receptors that when the neurotransmitter binds on and the metabotropic receptor is activated, this G protein breaks down, but the action of the G protein is to pretty directly open that ion channel. So in this case, we don't see this second messenger system. We just see this little blue piece of the G protein opening the ion channel here. So this is still a slow response compared to our ionotropic receptors, but we'd expect that to be potentially a little faster than if we're creating the second messenger. So we can distinguish these two types by calling them the second messenger system, or for this one where we don't produce a second messenger, we might say it's directly coupled to this ion channel. So we would call this direct coupling. So questions here about G protein coupled receptors, which are also called metabotropic receptors, and this whole system. Because I know some of you covered G protein coupled receptors in the past, but I'm not sure all of you have. Okay. So for this, our key takeaways should be, we call these metabotropic receptors, involves a G protein, and they're slow. If we see a second messenger, we're talking about these metabotropic receptors. Okay. So this moves us on further into our postsynaptic neuron. So these receptors, we remember, are on basically the second side of the synaptic cleft. They're the cell that's receiving the signal, which we call our postsynaptic neuron. So once we have the action here of the ionotropic receptor or the metabotropic receptor, and right now we don't have to think about which it is, we're going to see basically graded potentials produced. We're going to see changes to the membrane potential of the postsynaptic neuron. That's one of the things that I want you to keep clear as you're studying for the exam, is when you're thinking about a presynaptic neuron and when you're thinking about a postsynaptic neuron, because sometimes people get a little tripped up there. Okay. So we talked about graded potentials before, but basically what we're thinking about is that in response to the neurotransmitter, in response to these receptors, we're opening ion channels. And then we're either going to see depolarization, which is making our membrane potential more positive, or hyperpolarization, which is making our membrane potential more negative, or we will actually also kind of group together with that if we stabilize the membrane potential at a pretty low value. We'll talk about that in a second when we talk about these chloride channels. Okay. So before we talked about excitatory stimuli and inhibitory stimuli. So that's basically what we're talking about here. Here we're going to come up with some slightly more specific terms, but they're really just the same thing we talked about before. So if we open a channel that allows positive ions into the cell, and those positive ions would be sodium or calcium, this is going to cause depolarization. So we'd call it an excitatory stimulus. And now that we're talking about the synapse specifically, talking about interactions at the synapse, we're going to call it an excitatory postsynaptic potential if we're referring to that depolarization in our second cell, which is our postsynaptic neuron. So this is a longer word, but it's the same concept we've talked about before. So our EPSP is a depolarization in our postsynaptic neuron. Our IPSP, which is an inhibitory postsynaptic potential. Remember, hyperpolarization, if we bring our membrane potential down lower, this makes it hard for us to get to threshold, right? We need to get to -55. We need to have a little bit of depolarization to hit that depolarizing threshold in order to fire off an action potential. So if we hyperpolarize, if we bring this membrane potential down, it inhibits or stops the firing of action potentials. So this is our inhibitory postsynaptic potential. So one of the main ways we think about this happening is if we open potassium channels, because potassium wants to flow out of the cell. If we have positive charges flow out of the cell, it becomes more negative. We'll also see that chloride ion channels are associated with inhibition of our postsynaptic cells, so inhibitory postsynaptic potentials. They work a little bit differently. Okay. Questions so far? Okay, so we got these new terms, our EPSP and our IPSP. They're associated with some of the ions we've seen before. We can kind of tack on some more ions here. And we can think about other ways that you could create a depolarization or a hyperpolarization in our cell. These are the most common ways, but anything that makes that postsynaptic potential, anything that raises the membrane potential in that postsynaptic cell is going to be considered an EPSP. Anything that lowers that charge inside that second cell, lowers our membrane potential, we'll think of as an IPSP. Okay. So these postsynaptic potentials are changes in our membrane potential in response to a neurotransmitter binding to a receptor, whether that's an ionotropic receptor, so the fast one with the ions directly flowing, or a metabotropic receptor, which is involved with the G protein, so a slightly slower response. Okay. And then we break that down. Either ionotropic or metabotropic receptors can create excitatory or inhibitory postsynaptic potentials. It's not like excitation is always fast, inhibition is always slow. So these are kind of two separate trees that can affect each other. So for excitatory postsynaptic potentials, we do kind of associate specific neurotransmitters with either excitation or inhibition. Some neurotransmitters do both, and it depends on the receptor, but some neurotransmitters are much more commonly associated with excitation or with inhibition. So specifically, if we're thinking about excitation, our most common neurotransmitter is glutamate. That's just the name of a specific neurotransmitter that does this. And for our inhibitory postsynaptic potentials, the most common neurotransmitter that causes inhibition is GABA. For example, another sort of correlation with medications is, I think that benzodiazepam, so benzos increase GABA, so they're increasing inhibitory postsynaptic potentials. So that's another sort of correlation that we can find there. Okay. Since at a synapse, we have one axon, one neuron connecting up to our second neuron, our presynaptic and our postsynaptic neurons connecting to each other, we can refer to specific synapses as excitatory or inhibitory. So what we would be saying is, if we have an excitatory synapse, we'd be saying that neuron 1, that presynaptic neuron, is releasing a neurotransmitter that causes depolarization in the postsynaptic neuron. So that would be an excitatory synapse. An inhibitory synapse would be the opposite. If our presynaptic neuron releases a neurotransmitter that creates hyperpolarization in our postsynaptic neuron, that would be an inhibitory synapse. And we're specifying this here because we are going to take a look at how we'll also see modulator neurons, and that'll be important as we diagram that out. Okay, so an excitatory synapse, if we're releasing a neurotransmitter that's more likely to cause depolarization, this means basically that we're making it more likely to fire off an action potential in our postsynaptic neuron. We cause depolarization, excitatory, we're getting nearer to that threshold. So that's just what excitatory is. Inhibitory, we're inhibiting action potentials. We're making it less likely to fire off an action potential. The reason for that is that we're doing one of two things. Either we're hyperpolarizing that postsynaptic neuron, so we're making it more negative, or sometimes we'll see that what we're doing is basically membrane stabilization at a lower membrane potential. And what I mean by that when I say membrane stabilization is that we'll see specifically one of the actions of chloride ions is that they like fight against any sodium ions coming into the cell. And I think we have a diagram that shows you what I mean by that. So these are both considered inhibitory. Okay, cool. So this is just a reminder of our different receptors, our neurotransmitters, binding, creating our membrane potentials. So here we can see we've actually hooked up a voltmeter to each of these neurons so we could read it. So here we have our ionotropic receptor. It's just an ion channel. Ion channel is allowing flow of molecules. And we can see that we have a little spike on the voltmeter. Does somebody want to guess why we might expect in this diagram to see this excitatory effect rather than an inhibitory one? Okay. What I was trying to remind you of with that question is just that our gradient for sodium is stronger than the one for potassium. So potassium is slowly leaving the cell, but sodium, as soon as you let it flow in, lots of it wants to flow in. All right. So then we see a potential slow responsive receptor or metabotropic receptor. So here we see it triggering an ion channel as well. So in this case, does somebody want to tell me why we also are seeing excitation here looking at this diagram with our metabotropic receptor? Right. So to figure this out, we would look here at number 7. What's going on with the actual channel that we are eventually triggering. So we can see it's a potassium channel, but if we look closely, we can see that what's happening is that we're closing a potassium channel. So when I said on this previous slide that we want to just make sure that we're focusing on whether we're causing depolarization or hyperpolarization / stabilization, is because we can also see these effects when we stop potassium from leaving the cell. And if we stop potassium from leaving the cell, if we decrease membrane permeability to potassium, that means that the sodium that's still flowing into the cell kind of overtakes it. So we'd see a peak here, even though what we did was close a channel rather than open one. Okay. So those were 2 excitatory synapses, 2 examples of how we might have an excitatory synapse. So here we can see that we've hooked up our voltmeter to 2 new cells but we can see that what's happening at each of these cells is inhibition. So inhibition, we're thinking hyperpolarization mainly. Membrane stabilization also counts. So these inhibitory synapses are causing an IPSP, an inhibitory postsynaptic potential, which means that if we hook up this voltmeter, we'd see it go down. We'd see it become more negative. Okay. So the main ways we're going to see this happen is if we open either a potassium channel or a chloride channel. The potassium channel we've talked a lot about before. So when potassium channels open, potassium moves out. We're taking positive charges out. That means we're creating hyperpolarization. This is inhibition. The action of chloride channels is a little different, partially because we have different things going on with chloride concentration in different neurons. So it's a little more complicated, which is part of why we haven't been concentrating on it. So the big points about chloride channels is that one thing we can have happen in some neurons is that we have a chloride channel open and chloride moves in. We're bringing in a negative charge. So we'd create hyperpolarization and we'd be creating an inhibitory postsynaptic potential. So that's one thing we can see happen. And specifically, we might notice, right, chloride ions are negative. Our resting potential, also negative, right, negative 70. So if we think about those two facts together, we might be able to deduce why chloride is moving in here, which is that there's a strong chemical gradient in some neurons for chloride to enter the cell. And specifically, those are neurons. Here we can see in this picture, actually. Specifically, those are neurons where we use ATP to actively pump chloride out to create a chloride gradient. So that's this sort of simpler action of chloride we would see is if we create a chemical gradient for chloride, which some neurons do, then when we open a chloride channel, it flows in. The other thing we'll see with the chloride stabilizing membrane potentials, is there a picture? Okay, there is eventually a picture. Sorry, it's not the next one, guys. So this is the slightly more complex way that chloride can inhibit the postsynaptic neuron, which is that basically we want to look at this picture with neurotransmitter 1 and neurotransmitter 2. So some neurons aren't pumping chloride, so they don't really have a concentration gradient for chloride. So nothing happens if you only have one neurotransmitter because they're already at equilibrium, so nothing would happen if you opened a chloride channel. But the reason we'd still consider chloride inhibitory in those neurons is what happens if you have a second neurotransmitter act on that postsynaptic neuron. Because what we'd see in that case is if we have this neurotransmitter 2, say it's opening a sodium channel. So usually if we ignored the central part, just look at neurotransmitter 2, say it's letting sodium into the cell, we'd expect that to cause excitation because we have positive charges flowing into the cell. So we'd expect a depolarization. But you can see here our little voltmeter is flat. And the reason for that is the electrical force on chloride. So as neurotransmitter 2, this positive ion coming in, sodium coming in, we would start to raise the membrane potential. But instead, because we have a chloride channel open, those negative chloride charges are attracted to these positive sodium charges, let's say. So every time a sodium comes in, we'd also have a chloride come in to balance it out. So basically this disruption of the membrane potential with these positive ions creates a force on the chloride that will balance it out. So that is what I mean when I say membrane stabilization using chloride ions. So we'd still consider this inhibitory because it still makes it less likely to fire off an action potential, because basically it's turning off the effect of this excitatory neurotransmitter. Okay. So that's the stabilizing of the membrane potential. Okay. So we could have a pretty basic flow of our postsynaptic potentials. So our postsynaptic potentials are graded potentials. They're just specifically graded potentials in the second neuron in our chain, in that postsynaptic neuron. So a thing that we could see happen is that if we have lots of action potentials coming down our first neuron, down our presynaptic neuron, that would release a lot of neurotransmitter, because every time that action potential gets to the axon terminal, would let calcium into the cell, neurotransmitter would be released. Okay, so if we have lots of action potentials coming in, we're releasing lots of neurotransmitter. It's going to have lots of effects on our postsynaptic neuron. So when we have more neurotransmitter binding to receptors, depending on what type of postsynaptic potential we're talking about, depending on whether we're talking about an excitatory or an inhibitory synapse, we're either going to open or close channels, which is going to increase or decrease ion permeability, which will lead to greater or lesser ion flux. And you'll see that this is open or close, increase or decrease, greater or lesser. So if we're tracking a specific ion, any of these effects could lead to greater hyperpolarization. Or if we were tracking another ion and took another sort of path through this, we could lead to depolarization. So like to lead to hyperpolarization, we could say that we open potassium channels, we therefore increase potassium permeability, we increase the potassium flux, specifically the potassium flux is outward, leading to hyperpolarization. The other way we could get to hyperpolarization, right, is if we close a sodium channel. If we close a sodium channel, we decrease sodium ion permeability, which means we decrease sodium ion flux, which could also lead us to hyperpolarization. So you can track different ions using these. You could also track ions leading to depolarization. So I don't want you to think that every postsynaptic potential is hyperpolarizing. It just depends on which one you're tracking, whether you're opening or closing channels. Okay. We are at the end of our class period, so we are going to stop here.
So Monday